That's cold.
Yeah.
Yeah.
Right. Hello everyone, welcome to Lay It Out, the No Bullshit Science podcast.
With extra bullshit.
I'm Curie.
And I'm Alice.
And we're coming to you from our office.
We have a new location today.
We do.
Very exciting.
We normally record at Silent Demelza's house, but today we're in our office. And we're hoping this will become the more permanent location. And it might sound a little bit different.
Even though we'll miss Silent Demelza's cats.
We will.
Our favourite podcast supporters like to jump on the table mid-podcast and scare our guests.
Yeah. So yeah, how are you, Alice?
I'm good. I had a busy day today.
You did?
Yeah, I was being a volunteer for a research study.
Mm-hmm.
It's very exciting. Although I kind of was a very failed volunteer, which doesn't bode well because my logic about volunteering for studies is that if I volunteer and I'm a really good participant, then the karmic laws of the world will send good participants my way.
But you've also been a good participant in 5 studies this year alone. So I think you've got plenty of good participant
I haven't actually participated in all 5 yet.
Okay.
I will have by the year's end.
There you go.
Maybe actually more by the year's end. The 5 that I counted will take me up till like middle of June.
So then you could do 10 this year.
I could.
And that's 10 commas.
Oh god. So 10 good participants.
Out of 87, that's something.
Oh god.
Yeah.
Anyway, yeah,
So-
today was fun. I got to do, I just love doing them because you get to find out all these really interesting things about your health.
Yeah.
And then you get paid.
That's true.
And you kind of just get to sit there and people like make fuss of you all day.
Yeah. It's really unfair though because we have loads of brain imaging studies in my side and I can never do them because they're either looking for men or they want people that are right-handed. And
What?
I'm a corrected left-hander, so I do some things with my right hand and some things with my left hand. So I don't count as someone who's right-handed enough.
Okay, wait, I have several questions.
Okay.
Number 1,
Yeah.
why do they only want men?
I don't know, because the world loves men.
Oh, fuck's sake.
Yeah.
Number 2, I did not know you were a corrected left-hander.
Yeah.
Why did they correct you?
So my parents corrected me because it's something to do with the way that the Korean characters are written. It's really difficult to do it with your left hand.
Oh shit.
So they wanted me to learn to do it with my right hand. So I learned to write with my right hand, but there's still things I do with my left hand. So I've realised when I analyse what I do, anything that requires detail, I'll do with my left hand.
Okay.
So things like my eyebrows.
Precision
Yeah.
activities.
Or chopsticks, or if I'm colouring something in, I'll
shit.
generally do it with my left
So you can
hand.
stay inside the lines.
Maybe.
Nice.
But with writing and scissors and knives, I do it with my right hand.
Hang on a sec, because you don't need to be precise with a knife.
Not at all. If I'm hacking away at a steak, I'm not going to be like, right, this surgical line over here.
Okay, I'm glad you're not a surgeon. That's all I'll say.
But I'll probably scalpel with my... No, do you know what?
You probably scalpel with your right.
I probably would. Okay, fine.
Oh,
it's
God.
all the... I wanted to be a surgeon as well. Okay, it's probably like the small, fine, detailed stuff. Maybe my left hand is my more creative hand. Because it's eyebrows and colouring.
I love how your hands have taken on their own personalities
do.
now. They're like, today my right hand is moody, my left hand's really happy, so they're not getting on too well. So if your right hand's happy, you're just feeling more creative?
No.
Oh, because it's your left hand that's creative.
Yeah.
Okay,
with
sorry, I'm
it.
getting confused with the hands.
Left hand creative. They're not all the same,
okay,
you know.
Okay, I'll remember.
All hands matter.
It's like when twins don't have an identity.
Yeah, exactly.
Two hands.
Exactly. They always refer to them as the twins. You don't refer to my hands as the hands. That's the left hand and the right hand.
Okay, I think your hands need to go to therapy with you along with yourself.
Okay, fine.
Okay. You didn't let me tell you about the rest of
Oh,
the
sorry.
study.
Go for it.
Yeah, I'm wearing a glucose monitor at
You
the
are.
moment.
You look well lame.
Yeah, no, I didn't. It's cool. And it's going to tell me about my responses to foods. I'm really excited. Although I thought they were going to give me a scanner so that I could just, like, you know, eat an apple and then see what it did to my glucose. But they don't give us scanners.
Wait, I didn't actually finish my brain thing.
I don't really care.
Okay, fine.
How's your week going?
It's alright. I had an interesting start to my week. So recruitment for my studies has been going a bit slow. So on Monday, what we did is we got a spot in Croydon's Surrey Street Market, which is an old school fruit and veg market, which is where they actually used to film The Bill.
No way.
Croydon's big claim to fame.
Oh shit,
Yeah.
my granddad would love that.
Yeah. So the organisers there let us have our own stall where we would recruit people. So I'd send off my master students to Flyer, and all these people would come talk to me. And it was really great for the most part, except for the fact that Croydon has some crazies.
Of course. As does most of London.
Yeah, so one of the first people that came and spoke to me, he was this old man. He was probably in his 70s maybe. And he comes up to us and asks what it's about, so I tell him. And then he's like, you know what? Do you know what the problem is? I was like, what? What is the problem? And he's like, you're all just trying to make us feel ill. And I was like, sorry? And he was like, yeah, all you lot, you're all everyone, doctors, everyone, they're just trying to make old people feel ill. I'm fine. My brain is fine. And all this. And he just went off on one. And I'm there like, okay.
Except he's big into his conspiracies.
Yeah, which you'd think that I would be really into, but except he was attacking me.
Oh, no.
And then he was like, you know what? Do you know the reason why old people don't remember things as well? And I'm like, go on, educate me. Tell me.
Please. I want to know.
And then he was like, it's because we know so much more than you. I was like, oh, okay. And he was like, what's the capital of Germany? And I was like, Berlin. He was like, it would have taken me way longer to think of that. Do you know why? I was like, at this point I was starting to get a bit annoyed, so I started calling him sir. And I was like, why sir? and
I love how that's how you express your annoyance by like...
I'm getting passive at this point. And then he was like, it's because I have so many more memories than you in my brain, so it takes me longer to get them. I'm just there like, okay.
I'm not sure about that logic.
Yeah, and it just got to the point where I was like, he went on for so long about how old people are fine, and I'm there like...
But actually his argument is not very sound, because actually that's the reason why they are probably a little bit not all there. Like my granny comes up with these theories sometimes too. Like one of her main ones is that she thinks that only people who are really, really intelligent get Alzheimer's disease.
Oh god.
Yeah, because she's like, they're so smart that then their brain just like...
And it just got to the point, and I was like, okay.
That's her theory.
That's fine. You know, that's okay. That's fine. In my head, I'm like, you fucking came and spoke to me.
Did you have any more annoying
I
participants?
had one more participant that I wanted to bring up. So,
oh wait, they're not even participants, they're
interested
volunteers.
people. So this is two men. They were walking along, and then one of them looked interested, and I was like, oh, come and speak to us, blah, blah, blah. And he was like, oh, no, I'm too old. And I was like, no, we want over 60. And he was like, yeah, I'm too old, I'm 65. And I was like, no, we want you. And he was like, oh, you want me? And then his friend was like, what do you want me for? Sex! And I was just like... I was so horrified. And also, I fucking, you know me, I love an outrageous moment. But that was like, oh my god.
Like you're trying to recruit like people's grannies and granddads and you're getting...
I'm getting...
asked for sex.
Not even asked for it, asked if I'm going to offer it. Yeah.
They're picking up men, you're supposed to be working here.
Oh my
well, that
gosh.
sounds eventful anyway.
Yeah.
Did it go well though? Did you get many volunteers?
It did go well. We got quite a few people booked in.
Nice.
And a few people contacted us afterwards. So today we have two friends of Silent Demelzers again. We have, do you prefer Dave or David?
Dave.
We have Dave and Tony.
Welcome.
Hello.
Hello. Thanks for having me.
It's like now we give you the chance to talk and it's like, hi. Hi. Where are you? I was going to say, where are you coming from? But that's just because you have a suitcase with you. One of our guests has joined us the whole way from outside of Brighton.
Yeah, I've come from Brighton today. I'm moving in.
Oh, lovely. To the building.
Yeah.
Yeah, wonderful. What do you do in Brighton?
So one of my clients is out there.
Mm-hmm.
And so I've been working there for the last 3 months.
Lovely.
And I'm a technology consultant.
Oh, so that's why when you were asking Demelza if what her laptop was. Is that
partially?
Because you're a techie?
No, I just like that laptop.
Oh, okay.
Because he's techie.
Because
Yeah.
So
Yeah.
what is a technology consultant? Is that what you said?
That's what I said, yeah. Yeah, so I'm a technology consultant in the water industry, which is also why I was so interested in Demelza's borehole.
Wait, I think we need context. We need context.
We need to clarify.
Because...
By the way, he just said borehole.
Yeah,
just in case anyone didn't get
which could be associated with water in 2 different ways.
That actually did sound like extremely dirty. Should we explain what a borehole is, or am I the only person who didn't know?
I quite like the perverted image of David loving to hear about this borehole. Yeah. Okay.
A borehole is basically just a just a hole in the ground that water companies use to abstract water.
Mm-hmm.
And so when you get clean water from like in your taps, it often comes through a reservoir or a borehole and a couple of other ways. So it's just one of the ways that water companies use to get water out of the ground.
Okay, cool.
Wait, so what does your job actually involve?
So it varies. So we do what's called business integration between, say, big applications which companies use and the business itself. And currently I'm working on a piece of work where we're defining their strategy, what they should invest for a 5-year period. And that's everything from what computers you have, so like laptops and phones and that kind of thing, to big applications that do everything like HR stuff, payroll.
I get the feeling you like your job.
Sometimes.
Okay, interesting.
Because it is difficult because I'm 4 days a week I won't be in London, so I won't be at home. So then that can kind
lots
of wear
of travel
thin after kind of 3
I'm
or so
guessing
years.
hotels get old after a while.
Yeah, one of my friends an accountant, and she used to always be on with clients in different parts of the country.
Exactly.
And she hated hotels.
Yeah.
I used to be like, yeah, just stay in another one.
Do you know what you are? You're the guy that, you know when when you have a hotel breakfast buffet, right? If you don't go to hotels that often, you go in. Right? It's breakfast and you're there having like a 5-course meal of meats and then egg and then cereal and then a yogurt. And
At
then
least
fruit.
5 plates.
Yeah, and then the weirdest combination is that you'd have a cheese board for breakfast because why the fuck not? It's there. Yeah. But you're the guy that goes there and has just a bowl of cereal.
Yeah, exactly. I'm also the guy that when I leave, they go, "See you next week, Mr. Evans."
Oh God, they know, yeah. Oh, that's nice. They're like your family.
Yeah, we're like that.
So Tony, what do you do?
I'm a civil servant. I work on the tax.
Lovely.
Yeah,
You're a
great.
tax man.
Yeah, pretty much, yeah.
I actually know a lot of people that work in tax, but all at home. I don't know anyone here.
Is your job fun?
Yeah, it is at the moment, but you can rotate around quite a lot in the civil service.
Yeah.
So that's the good part of it, and it's good for you get to travel and stuff to some places, and it's a good opportunity to move around.
So what do you actually do?
We look at tax policy and sort of do research into that, and then we just help out and sort of advise on how we can change that, and also compliance and not what's not working at the moment and how we can sort of plug the gaps. It's quite good.
Nice.
I think this is the first episode in a while that we've had guests that are so different from us.
No, our last few episodes, all our guests have been low-key, actually scientists. They're supposed to be lay people.
Yeah.
But you guys are legit non-scientists. This is great. Although tech, what did you do in... Actually, let's move on.
Yeah.
What did you do in uni?
Natural sciences.
Ruining it. Okay, yeah.
You did natural
Yeah.
science.
Oh, they all did natural
Wait, so
sciences.
are you uni friends with Demelza?
Yeah.
Oh, okay.
So do you guys know Rob as well?
Yes, we do.
Yeah.
A previous guest of ours.
Yeah. Wait, so you had a pretty good relationship with science then?
Yeah.
Let's cut this bit out.
Which bit of the natural sciences
We're losing all our credibility.
were you into?
Well, chemistry and physics.
Okay.
More chemistry.
Yeah, and I was physics.
Okay,
More
it's fine.
chemistry,
They've got nothing
maths.
to do with what we do.
Cool.
Yeah,
Biologists.
No biology.
Zero biology.
Brilliant.
I
Wonderful.
didn't even do A level. I didn't really do it at GCSE.
Yeah.
Okay, cool.
So
Okay.
no idea.
Okay,
So we're fine.
This works.
Yeah.
Okay. Yeah.
Alright, should we get to some science then?
Yes, we shall.
Right, so Alice is going to go first this week.
Okay, so we have one guest who's listened to all of our podcasts and another guest who has listened to none. So I'll give a brief introduction to what I do. You will already know this. So I'm a nutritionist. So I mainly work on the interactions between your diet and what it does to your gut health. Specifically the bacteria that live in your gut. They're really important and they do loads, and there's loads of research in them at the moment with all these links to the health in other parts of your body, so not just in your gut. Like they're associated with your mental health, your immune system, loads of stuff. So this paper that I'm going to introduce to you guys today, so it's called the low FODMAP versus regular rye bread in irritable bowel syndrome, a randomized smart pill study.
I freaking love rye bread. I'm on a really big rye bread hype right now.
Yeah.
In Tesco, you know, in their fresh bakery, they have the bit of bread that's kind of nicer than the other breads that they do. They have this really nice rye bread. It's like really crusty, and the inside is super, super soft, and
And so they go into quite a lot of detail about the bread in this
nice.
paper. And it's sourdough rye bread.
I
I know. They wrote it down. I was like, I wish I could have been a participant in this study.
Nice.
Yeah. So this is a bit of a weird one because all research is in some way starts from the funding that you get.
Yeah.
Usually funding will come from like medical councils, where else does funding come from? Industry,
Charities.
Lots of different places. This study was funded by a bakery.
So funded by Big Bread.
Yes. But not even Big Bread. It's literally like Fraser Bakeries, which doesn't sound like...
I was going to say, what sort of bakery can afford to fund a clinical trial?
I've no idea, but a Finnish bakery apparently, because it's in Finland.
Well, those Finnish people.
But anyway, it's literally like a few scientists and a few bakers decided to get together and run a research study.
Nice.
What is low FODMAP?
Okay, good question. So low FODMAP, this study, as I read it in the title, was on people with irritable bowel syndrome, IBS. The low FODMAP diet is a treatment strategy for IBS. So IBS is a really difficult one to treat because not a lot is known about how you actually get it. It's a really big group of symptoms, and you don't have to have them all. So it's really varied in terms of diagnosis. So like 2 people can have IBS and they don't have any of the same symptoms, but they both still have IBS.
Okay.
So there's lots of different things that will probably cause it, but they haven't gotten to the root of... They've gotten to the root of some of them, but not a lot of them. So one thing that they've looked at in terms of treatment is this thing called the low FODMAP diet. And it basically is a diet that restricts certain types of fiber. So FODMAP stands for the types of fiber that you're not allowed to have. So these are fermentable oligosaccharides, monosaccharides, disaccharides, and polyols.
Okay.
So yeah, there's basically loads of types of fiber. There's loads of types of fiber in general. This is quite a big group of types of fiber. Yeah. So it is quite a complex diet to follow, and it's recommended never to just try and like pick up a book about the low FODMAP diet and just start it. You're always supposed to go to a dietitian and they'll give you specific advice on how you should go about it. Yeah. It's also not a long-term thing. What they say to you to do is, if you have IBS, you go on the low FODMAP diet for 4 to 8 weeks because it's really restrictive. There's loads of fruit and veg you can't eat, a lot of dairy products you can't eat, and there's a lot of research into the quality of it as a diet. Because there's a lot of people saying it could restrict other things. Which some things have been disproven, and they found that you actually aren't restricted in certain ways, but it's not recommended to be on it long term anyway, because it kind of gets in the way of your life a bit. So what they do is they put you on this for 4 to 8 weeks. Usually what happens is your IBS symptoms will go away, and then after the 4 to 8 weeks you start reintroducing small amounts of the high FODMAP foods and seeing what triggers you. And then you know kind of how much of it you can have and how much you can't have. So they're like, okay, have 1 tomato this day, have 2 tomatoes the next day, and then you see how much you can tolerate. And it's had quite a lot of success in reducing symptoms. I think it's like in terms of IBS, because any medical therapies and things like that have been really unsuccessful. So that's what FODMAPs are. Okay, so
what's the second? Because sorry, I didn't.
So we've gone through low FODMAP versus, so it was basically low FODMAP bread. So they made a bread that was low in all these fibers, and they compared it to a regular rye bread. And they gave it to people with IBS. And then the last part is a randomized smart pill study, which is a bit of a random inclusion there. Normally it would just be a randomized study. I'll explain what the smart pill is first, because it's really cool, and it's part of the reason why I chose this paper is because I'm actually using the smart pill in my study that I'm doing at the moment for my PhD. So the smart pill is a wireless motility capsule that you swallow, and you wear a monitor, and while it's going through your digestive system, it's like non-digestible, so you don't break it down. It just is a little kind of like specially designed thing to travel through your digestive system. And while it's going through your digestive system, it measures certain conditions, and then it sends that information to the little monitor that you're wearing. And then it passes through your digestive system, comes out the other end, and then you give us back the monitor, and we know all about what's going on in your gut.
Gross.
No, it's so cool.
No, imagine being the participant that has to fish through their shit that day.
No, you see, that's the great thing about it. You don't have to fish through your shit. You just flush it down the toilet.
What?
Yeah.
Oh, because you're the scanner.
The monitor.
Okay.
Yeah.
So I used that smart pill to analyse the findings in this study.
Yeah. So they used it to measure conditions of these people's gut after they ate the bread.
Is the smart pill well validated?
So it's quite new to being used in research. It was mainly used in clinics to begin with. But it is like the state-of-the-art method of measuring conditions in your gut at the moment. So the main outcome that research studies so far have used the smart pill for is transit time. So that's how long it takes for the pill to move through your digestive system.
Okay.
So in theory, it moves at the same speed as the food will move. So that's how we measure your transit time. Before, they used to use things like, they used to ask participants, or they still do use this, where you have to basically swallow a radio opaque dye. So you drink this drink and it has kind of like a slightly radioactive substance in it.
Yeah.
Right. Not dangerous at all.
Yeah.
Similar to living in Cornwall. And should I explain that? I want to tell everyone about my DEXA scan.
Okay, fine.
Okay, so I had to get a DEXA scan today as part of my my study participation, and as part of the DEXA scan, you get a little bit of radiation. But the lovely technician who was doing the DEXA scan on me told me that it's the same amount of radiation that you get while living in Cornwall each day, because Cornwall is so radioactive because of all the granite.
Maybe that's why you guys are like farming land. So the radioactivity is giving you like crazy good
Mutants.
crops and
crops. Giant carrots.
and then the cows are eating all your radioactive grass. They're getting all jacked up.
Yeah.
It's like the have you seen all the pictures of the mutant fruits and flowers and stuff in Japan after the nuclear bomb?
No.
Oh, they are crazy. Look them up.
Oh my god.
Yeah, look them up. They
Really?
are really crazy. Yeah.
What was I talking about?
Smart pill.
Smart pill. Oh yeah, and how they used to measure transit time.
Yeah.
So they used to give you a really small amount of this radioactive dye, and then they would
Oh.
get you to do an X-ray,
Yeah.
and they'd like measure
Perfect.
where it was and how long it took
Okay.
to get to certain points. So obviously a bit more, like, a lot of people didn't like having X-rays, and it's more, well, I don't know whether it was more expensive, because the smart pill is pretty expensive. And considering you flush it down the toilet at the end, it's like,
Yeah.
That's how they measure. And they measure other conditions too. So they measure pH and pressure. So like you have these contractions that go through your gut. So yeah, they use the smart pill for this. Not really sure why, because the study design does not really, anyway, that will become clear. Starting off, their aim was to compare the effects of the regular bread and the low FODMAP bread on irritable bowel syndrome. So this is what they say their aim was. Comparing the effects of regular versus low FODMAP rye bread on irritable bowel syndrome symptoms, and also to study gastrointestinal conditions with the smart pill. So they're kind of separate.
Yeah.
Did they say what symptoms they're trying to measure?
Yeah, so in the methods, they talk about how they measured symptoms. Normally in IBS, there's like quite a few validated questionnaires that are quite widely used and are quite easy to use. For some reason, this study just decided to make up their own questionnaire that wasn't validated and use that. So, questionable, number one.
Those Finnish bakers do what they want.
Yeah, I mean, they're baking their own bread, making their own questionnaires. This is how we do. So yeah, that's how they measured the gastrointestinal symptoms. And then obviously they measured the gastrointestinal conditions with the smart pill. And their other outcomes were, that was pretty much it. Yeah. So the study design, they recruited 9 people.
Good.
It's a great sample size.
Yeah, wonderful sample size.
There's not a lot of people in Finland. It gets quite cold up there.
9 people. They were also all female, which they don't give any justification for. But I suppose there might be, they don't go into this, but transit time and the condition or the functioning of your gut can be different in men and women.
Or
But
it's just girl power.
I don't really know. But I thought it was strange that they only recruited women and they didn't give any reason for it.
Yeah.
So they got 9 women with IBS. Also, they all had different types of IBS.
What if it is because, so I've realised from recruiting and just generally, women sign up to studies a lot faster and a lot more than men.
Yeah.
So maybe because they were only needed such a small sample size, it was just the first 9 women that came.
But here's the thing about the small sample size.
Okay.
So in their discussion, they talk about their power calculation.
Oh god.
Yeah.
How many were they meant to have?
Let me just find it. It wasn't how many they were meant to have. It was more how they calculated it. So, as I said, the aims were to assess whether the low FODMAP bread or the rye bread had different effects on their symptoms, and also to see what the smart pill measured, basically. But then they go and say that their primary outcome variable was breath hydrogen.
Okay.
And what they say they used breath hydrogen for was to measure colonic fermentation. So I mentioned your gut bacteria early on. What they do, and one of the mechanisms how you get IBS, or mechanisms of IBS and how it causes symptoms, is the bacteria in your gut. Their job is to digest fiber. They have different jobs, but this is one of them. So we can't digest fiber ourselves. So you absorb all your other nutrients in your small intestine. They all get like transported around your body. And then your fiber goes to your large intestine, where the bacteria have a party. Yeah. Because they're like, look at all this fiber.
The image I've got in my head right now. What? Do you know, like, finish the thing and then I'll say my image.
Okay, yeah. Sorry. So, the fiber gets to the bacteria and they use it and they ferment it. So we digest, they ferment. But it's kind of, for the sake of this description, the same. Right. So they use it to survive, but the products of the fermentation are really good for us. So they're things that can improve your gut health. And they travel off to different parts of the body and do good things in broad terms. But in people with IBS, this is kind of skewed. So they don't really know why. There's probably a few different reasons. But what happens in people with IBS is in regular healthy people, the fermentation causes the production of gases. If you don't have IBS, you're quite tolerant to this and it won't irritate your gut lining and you might just be a bit gassy and then you'll be fine. But in people with IBS, this causes, they say that they're hypersensitive, they have colonic hypersensitivity, and it causes lots of different symptoms, but things like quite severe pain or discomfort, a lot of bloating, and then obviously it can cause like diarrhea, constipation, all these different not pleasant things. Breath hydrogen, they're saying, is a measure of this fermentation. Okay. Which is questionable, first of all, because from what I can tell, it's not that widely used. This is the first time I've seen it as a measure of what's going on in your colon. Because if you think anatomically, gases that come out of your mouth
from your blood, right?
Come from your stomach.
Oh.
They'll come from your blood, but you know the way you've all that like loads of vasculature around your stomach. So they diffuse into your stomach and come up that way. Yeah. But gases from your large intestine, which is like the lowest part of your digestive system, generally go out the other way. So to be using
they should have measured anus hydrogen.
Basically, yeah. Is what I would think. I'm not the expert on this, but
do they explain
it doesn't
why?
sit well with me.
Do they explain why they do that?
No. They do give some references to other studies that have used breath hydrogen, but when I PubMed searched it, the first paper that came up was from 1992.
Do you know what the image I have in my head of this breath hydrogen is you know when you've had a really heavy night and then you go into someone or someone you know has had a really heavy night and then you go into their room and the minute you step into their room it stinks of booze. It's like that, like the bacteria have had a little party in your gut and they're fucked because they've fermented everything. And then from your mouth, because your body is their room, right? And then from your mouth comes out these alcohol fumes.
Yeah. That's exactly what's happened. The bacteria just let their hair down. They're having a great time. So they used breath hydrogen as their primary outcome, even though they said that symptoms were their primary outcome. Yeah. So they're calling breath hydrogen a symptom, which is questionable. Yeah. So yeah. Okay. So they measured breath hydrogen. So you do your power calculation based on like a previous study that would have measured breath hydrogen, and then you use kind of the effect size that they saw to calculate how many you might need for your study. But they state that and this is more evidence in the fact that I think breath hydrogen as a marker of colonic fermentation might not be a thing. Because they say, as the primary outcome variable used in this study there was no suitable previously published data in order to power the study.
You can't just stick it in your clinical trial then without like...
Wait, no, they didn't just stick it. No, they became a bit creative. So what they did was the study power for this study was calculated based on a preliminary test that was performed in Fraser bakeries.
No, it makes sense.
Yeah. So in which healthy participants ate regular rye bread or the low FODMAP rye bread, and then they analyzed their breath hydrogen content for like 6 hours in a postprandial period, and then just decided that they measured the breath hydrogen, saw the difference, which was 25 ppm. Don't really know what that means. But that was the difference.
Yeah. Also anyone who doesn't know what postprandial means, it means post meal.
Oh yeah, postprandial, post meal.
I didn't know what that meant. You know. Did you know? Until really recently.
Yeah, because there's loads of postprandial studies.
Because you lot would always talk about these postprandial studies, and I'm there like, yeah, postprandial. Oh yeah. And then I had to go look it up.
Ding, ding, ding. Silent denial is a fact. Classic example of scientists not knowing what they're talking about.
I think it's just us mouthing off.
So apparently breath hydrogen does check out as a method of measuring colonic fermentation. So I apologise, but I still stand by the fact that they should not have conducted their own random study in a bakery for their power calculation.
Yeah. I really want to try Phaser bakeries now though, so it's working.
You need to go to Helsinki. That's where they are.
I'll go.
Yeah.
If I go on holiday to Finland, I'm legit gonna go search out Phaser bakeries and I'm gonna ask them.
They actually must be like, when I first read this.
Ding ding ding, silent denial's a fact. Phaser Bakeries is one of the largest corporations in the Finnish food industry.
There we
Yeah,
go.
so
It's big bread. I
Yeah.
told you.
it is big bread. Because when I first read this, it was like Fraser Bakeries, and I was like, oh, like a little bakery
Yeah.
called Fraser.
You also can't read because it's Phaser.
Oh, sorry, Fraser.
This
is going well.
You've just been promoting a different bakery.
Yeah.
Fraser's
Bakery, I love
happy.
it, yeah.
I can phase it. Okay, sorry, Fraser. I prefer Fraser. Can I just continue with Fraser?
Yeah, let's help the small man. Fuck
Okay.
big corporations.
So yeah, they gave all the participants it was a crossover design study, which isn't ideal for studying things like related to your gut microbiota because it takes quite a long time for them to change. So by crossover study, it means that they used each participant as their own control. So everybody would have had both the rye bread and the low FODMAP rye bread, but like 2 weeks apart.
Right.
And their methods were interesting. They ate quite a lot of this bread in one day. So for breakfast, they had 4 slices of bread.
That would be enough to give me IBS symptoms. That would constipate me so bad. What the fuck?
So for breakfast they had 4 slices with I think they said some vegetables, and coffee and tea. Oh, and some cheese, and they could have a bit of spread too. if they wanted. And then for lunch they had 2 more slices of bread, and that was 6 hours after breakfast. And then for dinner they had an additional 2 slices of bread.
These people are having 8 slices of bread a day.
Yeah,
Of
and it
course
was-
they're going to be constipation or have diarrhea.
So this is 10 to 12 hours after the breakfast. Yeah, they just used a specific type of sourdough starter in the low FODMAP bread that they said would have digested the specific types of fibre that are FODMAPs.
Okay.
So that's how they made it low FODMAP. So they had it 2 weeks apart. So they'd come in for one day and eat all this bread, and then they'd give them 2 weeks, and they'd come in the second day and eat all this bread. So I'm only very early on in my study, but we have had some smart pill training. They seem to have used it a bit wrong. So the way your digestive system works, and because the smart pill is quite large, we say the size of a large vitamin pill, but it is quite big.
Is it like cod liver oil size?
Maybe a bit bigger.
A bit bigger.
Oh,
Yeah.
shit.
Is that not a bit painful?
So it shouldn't be. You drink it with water.
Is it peristalsis when you?
Yeah, so it's there's waves that go through your digestive system every 30 minutes, the whole way through your digestive
Yeah,
system.
because if I eat a big piece of chicken
Yeah.
and I try and swallow that, that's quite
When you
painful.
I was going to say, why aren't you chewing your food?
It just happens. I'm not saying shovel it down, but when it just kind of slips down.
When you can't help it, it just falls down your throat. Yeah, exactly like when a piece of chicken finds
This is
its way
like,
into your
have you
throat.
guys seen that video of the guy when he goes fishing and the fish,
Oh, yeah.
the fish flies off the hook and literally just slides down his throat?
I mean, he chokes, doesn't he?
Yeah, it's so weird. It's like a proper fish, and yet it just slides down his throat and it's just stuck there. It's like that.
So,
Anyway.
not as big as a fish,
No.
probably a bit smaller than a piece of chicken, but bigger than the cod liver. So anyway, now that we've clarified the size of the smart pill.
We haven't really.
No, we haven't. But anyway, we're moving on. So the smart pill, we've been told, how I've been trained to use the smart pill is that in your digestive system, about every 4 hours, you have these really strong contractions that go through your whole digestive system if you haven't eaten anything. And that's when you're really hungry and your stomach rumbles, that's what they are. So it's your stomach, your body's basically looking for food to digest, is how I think of it. And when you take the smart pill, the standardized way to take it is you have this thing called a smart bar, which is a special bar that's been designed to standardize the delivery of the smart pill. So when my participants come in, I'll give them the smart bar. They'll eat it. Then they'll take the smart pill, and then they're not allowed to eat for 6 hours. So that allows enough time for the food that the smart pill or the smart bar to kind of pass through their system, and then also for their stomach to be empty for long enough for these really strong contractions to go through their digestive system. And what that does is it pushes the pill from your stomach into your small intestine. So you need the strong contractions to do that.
This sounds terrible.
You don't
You
feel any of
just
this.
have to sit there and be hungry. That's
Well, no, 'cause you've
literally my
eaten-
worst nightmare.
You've eaten the bar.
So what does the bar do again?
It's just a way of standardizing.
So they've all eaten the same.
Yeah.
thing before.
So, and I guess because you can't expect participants to, if they've eaten something different, if they all eat something different that morning for breakfast,
Yeah. Okay.
it might react differently. It might take longer time to clear from your stomach, so the pill might take longer to move on.
Okay.
So it's just a way of doing that. So what we do is we ask them to fast from 12 hours before their visit, so they've nothing, everything should be moved by then. And then give them the smart bar, so they're all starting from the same baseline. And then the pill, and then it should all just hopefully move similarly.
Okay.
So that's what they're supposed to do. But in this study, they gave them the smart pill, and then they had fasted from the night before, gave them the smart pill, and then got them to eat the bread. They don't really say whether the cheese and fruit and veg and coffee and tea and stuff that they had, whether they all had the same amount,
Yeah, okay.
or whether they had different amounts. Which leads me to believe they might have just been able to have as much as they want. And then they had an issue later on, which was that the smart pill, actually, I'll get to that when we get to the results. It all becomes clear.
Oh, suspense.
Yeah, go on.
Were they allowed to put milk in their coffee or tea?
Doesn't say.
Okay.
But considering they're letting them have cheese, I would say so.
Okay.
Does it say what sort of tea?
No. Were you wondering, is it chamomile?
Is it chamomile? Or perhaps peppermint?
Like the chamomile that you're enjoying with your wine right now.
I'm pretty wild.
And I'm guessing they haven't said if they're restricted on what sort of cheese you can have either.
No, just cheese. But isn't that a really like cheese? That's like a continental breakfast, what they're describing.
Yeah, but like,
They all eat cheese and bread.
and
we're in Finland.
Depending on what sort of cheese you have, it's going to affect you differently. For example, if you're lactose intolerant, you can't really have soft cheeses, but hard cheeses are okay.
And it's also not, exactly, it's not telling you also whether everybody ate some of each of these things, or whether some people just had the bread and spread, or some people just
Bread and
had
spread.
the bread and cheese.
I like that.
Yeah, it doesn't say. Anyway, so yeah, they gave them this after they had the smart pill, and then they went home with their monitor for their smart pill, and they recorded their symptoms and also their breath hydrogen every 30 minutes until 6 hours after. So they would have stayed around until 6 hours after their dinner, and then they sent them home, and then got them to fill out more of these symptom questionnaires until the smart pill had passed through their system. So we can move on to the results now.
Yay!
The exciting part. Their first result was that breath hydrogen was lower following consumption of the low FODMAP bread.
Isn't this one big fat duh?
Yeah,
Because if it has non-fermentable fibre, then duh.
exactly. Wait for more of that. So, yeah, lower in after consuming the low FODMAP bread than it was after considering the regular bread, which is not very surprising because it doesn't have any of these fibres it does actually have some, but really low amounts in comparison to the regular rye bread. The second one was there was no change in GI symptoms because they were just giving them shitloads of bread.
Well, yeah.
Yeah, but they did say that there was a suggestion of decreased flatulence after the low FODMAP
Well,
bread.
duh.
But it wasn't significant, so we can't actually say anything about that. But they wrote it down anyway.
Yeah, I mean, to be fair, if I thought I was eating less fibre, I would probably think I was farting less as well.
And their questionnaire wasn't validated.
I wonder how they asked it.
It was a visual analog scale. So a visual analog scale is a line. It's actually just a line on a piece of paper. And it's a certain length, and it's the same length on all the pieces of paper. And it says things like, so say some of the symptoms were, so say they had a symptom of abdominal pain. Right. So it would have said, how much abdominal pain are you feeling right now? And on one end of the line would have been the most I've ever felt. And on the other end of the line would be none at all. And you have to mark on that line. So it's like a scale. Okay. So if you're feeling like kind of in the middle amount of pain, you put a mark in the middle. If you're feeling like really horrible pain, you put it close to the most pain I've ever felt. And then they would have gotten them to do this. They were getting them to do this every 30 minutes after. So then you can kind of compare. And they use these kind of scales for a lot of things. Like we'll use them for hunger.
I would have got them to count how many times they farted that day.
Yeah. Did they do this for flatulence? So if 1 is not fart at all and 10 is the most flatulent I've ever been.
There's just one constant wind coming out of your asshole all day.
Constant, non-stop flatulence. No flatulence at all.
And also, right, so like you have your level of fartiness can vary per fart, right? So you have one fart which is just like a big fart, and then you have those other farts that are like
loads of little farts.
Yeah. How do they count?
It should be like volume of gas, right?
Or
I mean, I don't know, maybe they'll invent some sort of contraption that you can like strap to your ass.
Or maybe
measure.
they need like one of your glucose monitors, but one that you put next to your asshole.
Yeah, and it's gonna measure what's coming out. Perfect. Yeah, so honestly, the question is, I don't know. It just says flatulence and that they use a visual analog scale. But because their questionnaire is made up, we'll never know.
Okay.
So I'd say it would have probably said something like, the most flatulent I've ever been, no flatulence at all. And they just had to mark along the line.
If they used women as well, this whole thing really depends on where they are on the menstrual cycle. Because I don't know about you, but when I'm getting up to my period, I've got like
way more farty.
Digestive problems and way more farty.
How do our guests feel like that?
Yeah.
Right answer. Yeah.
And like periods notoriously mess with your digestion.
I know, yeah. They don't say whether they hang on, let me check in case I make more false claims. I'm pretty sure in the exclusion they didn't mention.
You get done for defamation by Fraser.
Don't, they're the biggest food industry in Finland.
We're not talking about Fraser, this is a different study. That was done by Fraser. They're a rival. Yeah, that was it with the flatulence anyway. And then they said that gastrointestinal pressure, so that would have been measured by the smart pill, correlated with symptoms.
Duh.
Thank you. Thank you. So this is well known in IBS that, as I explained earlier, the bacteria ferment, make loads of gas, and this is causing the symptoms. So groundbreaking.
This study is stupid.
Yeah. I think what they were doing was they had some funding and they just really wanted to play with the smart pill.
Yeah.
Because that's what I would do.
And make bread.
And make bread.
Promote themselves.
Yeah. Promote low FODMAP rye bread.
Because now I kind of do want to try Fraser Bakery's rye bread.
Yeah. Let's go on a trip to Finland.
Yeah.
But it only correlated with symptoms after the regular rye bread consumption, but not following low FODMAP bread consumption. But they said it was nearly significant in the low FODMAP bread consumption.
I don't get this study.
No. And also they literally said was nearly significant, which is not something you say. So then the final finding of interest, although actually no, there's one more big finding of interest that I'll talk about after, but it's only really of interest to me. So there was no difference in pH, transit time, or pressure between the breads. So this was all the things that they used to measure smart pill to measure. And the reason there was no difference was because they actually found that, remember earlier I was saying how they gave the smart pill to the people before. How they gave the smart pill to the people before the bread was a bit strange. So they actually found that the smart pill stayed in the participant's stomach for between 15 and 22 hours. Ooh. So it didn't clear. Because I'd say what I would imagine what happened. This is all hypothetical and me being...
You being a scientist.
Throwing my opinions out there. Which is very scary. But I would say that the bread probably took different times to empty, so they'd never got that time when it was completely empty, and they didn't get that really powerful wave that would push it through to their small intestine. So the smart pill was just sitting in their stomach for ages, and it didn't move with the bread. But they did suggest that the 1 day, 24-hour feeding period and the smart pill wasn't the best combination. They were like, it's better off to be used in longer term feeding studies, which is what I'm doing.
Ooh.
So yeah, watch this space.
But if anyone is 18 to 64
yes,
and healthy,
and are you pimping my study again?
And is a habitual snacker, which means you have more than 2 snacks a day, then email optigut@kcl.ac.uk to see if you...
So really the findings of that study, what they aimed to do, they did not do. And their findings were just that increased production of gas in IBS causes symptoms.
But now...
And that you should use smart pill in longer term studies.
But now you're all craving rye bread.
Okay, so what did you guys think of that paper? Any thoughts?
Lots of flaws.
Yeah. Great.
Yeah.
Wonderful. You're scientists now. Well, nutritionists.
Like going back to the smart pill.
Mm-hmm.
Does it not impact your transit time by kind of deliberately fasting and that kind of thing, or is it just not important? You just compare it to that same scenario?
Yeah, so that's kind of, I guess, links in what they're saying about it being short term.
Yeah.
So, yeah, you're right. The fasting and then eating that meal, and then them expecting smart pill to follow along with it, and that just be their one measurement of transit time. Yeah, it's more like the shorter period of time, it's more likely that things like fasting or what they ate before and this kind of thing could influence it.
Yeah.
But the hope is that in the longer term studies, because in my study, for instance, it's like a 4-week intervention. And what we do is we get them to take smart pill at the start. So they'll take it before they start any of the dietary intervention part. That's just to measure your baseline. And we do get you to fast the night before. But that is, as I said, in order to standardize things, so to make sure that it all does move the same way. So I suppose then the hope is that if fasting is changing their transit time, it's going to be changing everyone's.
Right.
Okay, let's go.
So the paper I chose today is completely outside of my field, so don't ask me any questions.
You can ask it as many questions as you want and make them as outlandish as possible.
This paper is really cool. I don't understand how they did a lot of it, so I'm not going to go into detail in that. But it is called the H-Iris-Plex S system for eye, hair and skin colour prediction from DNA, introduction and forensic developmental validation. So it's kind of nice because our
Lovely.
papers are kind of linked today because they're both products. Yeah. So mine is validating a tool that you could be used in forensic investigations of crimes and blah, blah, blah. At the moment, so when we have a standard forensic DNA analysis, so when someone goes to a crime and they pick up your like blood samples and hair and blah, 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 the main thing that we have right now is that they profile the DNA, they get the full genetic profile of that sample, and then they match it against either a known suspect or a database. So if you're not on that database, which most people aren't, although now with 23andMe, who knows, the chance of them finding their perpetrator is quite small. So what they need to do is, because that fails a lot, they're going into what's called forensic DNA phenotyping. So this means, this is like a big term for any intelligence approach that can provide information about someone's physical appearance from their DNA. So externally visible characteristics, which is what a phenotype is. The main things that at the moment this forensic DNA phenotyping can give us is someone's biogeographic ancestry, so what people use 23andMe a lot for. And another reason why a lot of people use it is to get physical appearance from remains. So this could be from if someone's missing and you find some skeletons, you could get their appearance from that. Or if there's a mass disaster which has loads of victims, you can sort of get a picture of what they might look like. So the fields that use this forensic DNA phenotyping a lot is anthropology, so study of humans and human behaviour, evolutionary biology, and paleogenetics, which is the study of the past with genetic material from like ancient organisms, so from dinosaurs, skeletons, and blah blah blah.
And all those big mammals that used to live in Australia
Yes.
that we killed off.
Over the last 10 years or so, there's been loads of these huge studies which can look at your entire genetic makeup of a big population of people. And they've established various predictive, what are called SNPs. So these are single nucleotide polymorphisms. I made lots of notes on these because that's the big words that we have in this one. And from that, they have these predictive SNPs. They have managed to establish lots of external visible characteristics that they can predict. So what a SNP is, it's the most common genetic variation that we have. And a SNP is a difference in a single building block of your DNA. And one of these building blocks is called a nucleotide. So when there's one of those that's changed, that's a SNP. They occur normally throughout your DNA, and there's about one per every 300 blocks. So within one person, you have about 10 million SNPs. So a lot of genetic mutations are random, but a SNP isn't. A SNP is inherited. And it's maintained at your sort of gene level throughout your generations. And it's one of the reasons why organ transplants get rejected quite a lot because your SNPs don't match that stranger's SNPs,
Right.
and it rejects it because it can't take it on.
Is that the technical name? That's SNP.
A SNP, yeah.
Oh, right, okay.
We love acronyms.
Yes.
Yeah.
And abbreviations.
Yeah. So overall, in a single population, certain SNPs will be present in that population, and it's because it's not random, and as a population evolves over years and years and years, it all starts to mix. So over time, it will sort of even out, and you'll generally have very similar SNPs within a certain group.
Yeah.
That's why we can sort of, pinpoint certain SNPs to be risk factors for certain things, because you'll have this one difference, and everyone that has this certain trait might have that one difference in their thing that they don't normally have compared to the rest of their population.
In their thing.
In their thing,
Yeah.
Very scientific terms here.
Thing is potentially the most scientific term that you'll ever hear.
No, it's thingy.
Thingamabob.
Yeah. Do you know my sister has this habit of when she talks to me, she'll be like, "Oh, you know that thing, that thingy, and I'll be like, "No," and she's like, "You know, that thingy." Anyway, so these human pigmentation traits. So this includes hair, skin, and eye color. We know a lot of those SNPs now. So at the moment, there are pretty accurate forensic tools that exist to predict hair and eye color, and this is because they have evolved in large populations of the very similar sort of people. So in a European population, they will generally all have a certain eye color, so mostly blue or green eyes, and that doesn't vary a lot in a population. So things like eyes and hair are generally quite consistent, so it's been really easy to find the SNPs for those. Although that's very simplified. Things like blue eyes are actually really difficult. They still don't know exactly how blue eyes happen, but yeah. But skin colour is really different because apart from populations that are really concentrated, like South Asians or certain parts of Europe, there's so much variation that goes on globally that skin colour is really, really difficult to find, because these studies where they found these SNPs rely on these huge populations of similar people.
I have a question.
Yeah.
You know the way that you said that they can't find out how blue eyes occurred?
Mhm.
So is the whole thing of you having one blue eye allele from your mom and one blue eye allele from your dad, and then you get two blue eye alleles? Is that false? Or is it at a deeper level than that?
I think it's at a much deeper level than that. Because do you remember we sat through that talk of that guy who did the whole genetics of blue eyes?
But I remember he explained blue and he explained brown, but he didn't explain green. Because I remember feeling discriminated against. Because I was like, I have green eyes, why isn't he explaining green?
Oh, maybe it's the green then. Which would explain why your results are so, vague.
Yeah, okay, so green is just, really light brown. But it is green.
Yeah.
Like, my eyes are green.
Yeah, your eyes are very green. Okay, so what this group did is they were like, this cannot happen. They've already developed something that's really good for hairs and eyes, and I think the tool that this group developed is used quite a lot. And now they're like, right, we want to do something that helps you predict skin as well. So most skin studies right now have limited reliability because they've generally had a really small sample size. So this group really tried to change that. So they developed this prediction model that they're calling, which is the HIRIS-Plex multi whatever the shit I said.
Right, yeah.
Which has 5 skin color categories based on 36 SNPs from 16 genes. So these SNPs are usually found near or in genes, so that's why it's from specific genes. So it's 16 genes and there's 36 SNPs in and around them. And the 5 skin color categories are very pale, intermediate, dark, and dark black skin.
Ooh.
Now, I don't know what you think, but I don't think this is everyone's skin colour, because we're the yellow mentioned, because I'm not here.
I was about to say, let's go around the table and say what we think we are, and then everyone else can say what they think we are.
I think I'm pale.
So wait, what was this?
I'm not that pale at the moment, though.
I think, would you
Which was my other thing I wanted to say about this, but yeah. Maybe
intermediate?
Actually, because I'm not, Mediterranean skin.
But would Mediterranean be intermediate, or would that actually be pale? Or would it be dark?
Because compared to a person from Africa.
But they'd be dark or dark black if they were.
Exactly. Yeah.
Yeah.
Why is it like pale and then it's not like white. It then just becomes dark black.
Yeah, like they go from pale. Yeah, you're right. They're not
Yeah.
using the.
There's no colour mentioned at any
No.
point
But dark black.
until dark
Yeah,
black.
yeah.
I don't know what to think about that.
No, it's a bit.
Wait, where are these people from? No, my
Texas.
Yeah.
You can cut that out. Oh, there's pictures.
There's lots of pictures.
Ah.
Stop! No spoilers.
Oh,
We'll go to the
sorry.
pictures
Oh,
later.
Oh, sorry. Okay, can we do the table thing where we go around?
Yeah, what do you think you are? Well, at the moment you're quite tan.
I know, yeah. So then I'm inclined to say that I'm pale rather
Yeah.
than very pale.
You're not very pale. I've
I
known
don't think
much
I
paler
am.
people than
Yeah.
you.
Yeah.
I'm trying to
I have
think.
friends who are translucent.
When I buy foundation, where do I go?
Yeah.
Although if I was trying to buy foundation and there were 5 shades, I'd be pissed. I think I'm pale.
But then we're the same.
But we're both quite tanned right now. And also, this is the other complaint I had, because people have different undertones to their skin.
Yeah.
So there's yellow, neutral, and pink.
Correct. Yeah, pink.
And I am very yellow.
I'm probably pink.
You're pink. You would be quite yellow? Neutral? I think you're
Yeah.
quite neutral.
Yeah.
And then you would be pink.
I'm pink.
Yeah.
I'm gonna say I'm
It's
very
like, you
pale
know,
right
I
now.
feel like the people who are pink really know they're pink. Like, yeah, I'm pink.
Anyway, so
Wait, what about Demelza? We didn't say what she is.
Demelza is pale.
Pale pink?
I don't know.
Same as me.
Pinkish. I'm pretty
Judging by how red her face went when
Oh,
when Dave said that story earlier on, I'd say she's pink.
I'm convinced that Demelza has some sort of Mediterranean in her though.
Ooh.
Ooh.
Wait, what did her 23andMe say?
No.
Oh.
But she has quite a bit of Jew in her, so it's probably the Jew.
Oh, okay.
Am I allowed to say that?
I don't know. I feel like if you said Jewish, it might have been a bit better. But you're like Jew.
I think I'm gonna get in trouble.
The Jew.
That's right.
Well, it's up to you. You can take that out as well.
No, do you know what? I love this paper because it opens me up to so many racist jokes.
Can it give you proportions
Wasn't
of
it you?
ethnicity?
Who was it? It was you and Yara when we were out the other night, and the two of you were like, "Yeah, I love a bit of racism."
I do. It's my favourite genre of comedy.
Like, I was just sitting there. Me and Dan were sitting there looking at each other. We were like, "We can't."
Do you know what?
say
No,
that."
Dan's looking at that because he's jealous. He wishes he could say that shit.
But it's not like there's like like stand up, sketch, and
Racist.
racist.
No, okay, fine. It's my favourite subgenre of comedy.
And
It's
you
just-
like a lot of the Irish comedians. They go all out on that.
It's funny. I like stereotyping.
Yeah, you see, yeah.
I mean, I'm like, you know, it's funny. I think you should be able to make fun of yourself. And also, I make racist jokes about Asians all the time.
Fair.
So I'm
You do.
okay.
Like, nobody can touch me.
So I'm okay. And I have a black friend, so there.
A black friend who you went to see Black Panther with.
Yeah.
Yeah. There you go, you're fine.
Anyway, so then
Untouchable.
in my notes, I did write a section called Materials and Methods, where I was going to write the materials and the methods. But actually,
instead-
I started to write it, and then at the bottom in massive writing, I put lots of genetic stuff I don't understand. So I'm not going to go into it.
So you're not getting any methods. They decided that they were interested in this, and then they actually found these
I'll
results
tell you
on the
one
floor.
bit of method. The one bit of method that I did write down is that they collected body fluid and tissue samples in-house. So from their fellow lab members, they got
Oh, shit.
saliva, blood, touched items, semen, and vaginal swabs.
Hang on.
That is invasive.
So they asked their colleagues to have a wank
Pretty much. Yeah.
in the office.
Yeah.
God, that's very unhygienic, isn't it?
You think handing stool samples are embarrassing? Imagine handing over a copy of jizz.
Yeah, no.
Not being able to
that's-
make eye contact.
That's definitely worse.
Okay, so results. So these results are basically just loads of different ways that they try to validate their new tool. Because obviously they want to now get this tool out so that all these cool crime people can use it.
And they can get really rich and famous.
Yeah.
Similar to Fraser
But I think
Baker.
they're going to need a better name than H. iris plex S. What is it? H. iris plex S.
I don't think you've said it right once since we started this.
H. iris plex S. It's really hard. H. iris plex X. Plex S. H. iris plex S.
H. I said it like you. I don't say it that way. H. Irish Plex. H. Iris Plex S.
Exactly. Excuse me, Lakshmi Chaitanya. I think you guys need to work on your branding.
For a second there, I was like, whoa.
Never mind. I was about to say what language are you speaking? And I was like, no, don't say that.
Right, anyway. So results and discussion. So the first thing that they tried to do is they tried to see if their little system can tell the difference between if there's a mixture of samples. So what they did is they got samples from 2 individuals that have different pigmentation. So one person had brown eyes, black hair and dark skin, so it's a black guy. And one person had blue eyes, blonde hair and pale skin, so they're Aryan. And then they mixed up their DNA. And they had 3 different ratios. It was 1:1, 1:5, and 1:10. And basically what they found is that their little system, it could not tell the difference if they were mixed to equal ratios. But from 1:5 and 1:10 ratio, they could tell that there were 2 different samples in that thing, the mixture. Which is pretty good, because you're never going to get a pure DNA sample if you're swabbing a crime scene. So then next, they wanted to see if the results that you get can be reproduced by many different labs. So they used 5 labs, and they had 30 DNA samples. And all 5 labs managed to genotype, so get the whole genetic code, of 25 samples correctly out of the 30. Not bad.
That's quite good.
Yeah, that's all right. And then they wanted to... So then they gave them codes of genes. So they gave them a genotype, and then access to their prediction models, so of their eye, skin, hair colour. And out of them, they got 28 out of 30 right. So there was a 93.3% accuracy that they could get their eye colour, hair colour, and their skin colour right.
Wow.
Pretty good.
93%.
Yeah, that's not bad. Yeah. But I mean, you know, that does mean 7 out of 100 people are falsely imprisoned right now.
Yeah.
You should make a Netflix documentary about that.
A Netflix documentary?
Yeah.
I should. There's loads though. There's so many coming out.
That's why
now.
I said that, because there's so many.
No, like actual cases, not documentaries. Oh. Like, you know, there's so many people it's like stuff coming up of people that have been in prison for like 20 years, but because they didn't have the DNA tech back then, they got imprisoned. And then I was showing Jamal this guy earlier, who this would not work on. He went to prison for 17 years because the guy who actually did the crime looks exactly like him.
Oh, Jesus.
Yeah, like literally, they had pictures of their 2 headshots together, they're exactly the same. Obviously, you can tell the little differences, but it's the same.
Do you know did you watch that? It was like a series, but it was like a docu-series.
I love a good docu-series.
About the O.J. Simpson trial.
What was it called?
But apparently that was one of the first trials that they used DNA evidence in, and because everybody was like, oh, what's DNA, what's going on, that no one trusted it, even though it was like quite legit. And then he got away with it.
Oh shit.
Yeah. But it was because it was like really early on, and there was all these things about how they brought out the DNA evidence, and because they brought in a scientist to explain it to all the jurors, and that scientist clearly hadn't been on our podcast and learned how to explain science.
I mean, he was a scientist doing DNA in the early stages. I can imagine what sort of person he was.
Yeah. So everyone was just like, what is this guy going on about? Even though it was like such strong evidence.
Yeah, sitting there with his trousers up to his nipples.
Yeah, basically. and then they just, we've come a long way from there.
So then the next stage they were like, okay, fine, people can reproduce the same results. This is great. Is this specific to humans? So what they did is they got DNA from a dog, cats, rats, mouse, a pig, bovine, chicken, and a chimp. And they ran
bovine?
Cows.
Thanks. Jargon.
That's Demel's sort of jargon. And then they ran it all through this software. And except for the chimp, none of them raised any sort of signal in the thing. So apart from this chimp, which is a close...
And chimps are the closest to humans.
Exactly. So this closely related non-human primate is the only thing that sparked any sort of signal in their system. So again, pretty good. And then they wanted to look at, okay, so how does this deal with degraded DNA? Because when are you ever going to get a crime scene with fresh DNA? So they would get DNA and they exposed it to UV light. And they did it for 0 seconds, 30 seconds, 1 minute, 5 minutes, 10 minutes, 20 minutes, and 30 minutes. And they...
Surely they'd be a lot longer.
Exactly. So they... Oh, I should have mentioned that they had 17 SNPs that they were looking at for skin colour. So the full 17 SNP profiles were obtained from all samples up to 10 minutes. So anything that was exposed to the UV light for 20 minutes or half an hour, they couldn't get the full genotype.
Oh no.
Yeah. So,
and you're likely to be...
Unless it's done in a dark room with no windows.
Oh god.
You're kind of fucked. Yeah, you're going to have degraded DNA.
Did they compare it to the intensity
No.
of UV?
No
and
changes in intensity. Basically, if you want to commit a crime and you don't want to get your DNA done, do it in a tanning bed. That's all I can recommend.
One of the standard ones.
Or on a sunny day in August.
Yeah. So then what they wanted to do next is they wanted to use casework samples. So I think now they've just have random samples that they've tried to make as real as possible, probably from their colleagues' jizz. So they had 6 samples of mixtures and they wanted to, nope, that's a lie. They had 25 simulated casework samples. 90% of these samples were correctly genotyped and they were also correctly phenotyped for the hair, eyes and skin. And they also had 6 samples of mixtures of all this DNA. And 5 of these were correctly identified as mixtures, but they could not tell what the phenotype was. So from these mixtures, they couldn't identify what colour their hair was, what colour their eyes were, and what colour their skin was.
Nothing.
No. And then what they did is then they got a sample set of a much bigger population. Now this is genetic data that's available from bigger, huge studies, such as the 1,000 Genomes Project, where they're trying to get 1,000 genomes. So for this, they've got a sample set from a population called HGDPCEPHH952.
Oh, that's really snappy.
Yeah, I
Has a
know.
ring to it, doesn't it?
So from this, they had 952 people, or in their sample, and they excluded 190 from. So this was after they excluded 190 because they had a DNA shortage. And several of the 17 SNPs, so I said they had 17 SNPs that they had identified that would tell you their skin colour.
Yeah.
Several of these had really strong differentiation between different continental regions. So this sample that they have is a global sample.
Okay, yeah.
So between the different regions, they did find that there was strong differences between just different areas. So when they were differentiating these though, what they found was that it was actually quite hard to get them into their 5 categories. The most prominent differences were European versus non-European, Asian versus non-Asian, and African versus non-African. So I suppose if you had enough of a sample, you could whittle it down, but the chances of you getting enough samples to run it 3 times is quite slim. Also a lot of money. So then they used another data set as well. Now they had 1,539 individuals from 59 populations around the globe, and they then wanted to test their 5 categories of skin colour with these people. The global skin colour prediction achieved with the system agreed with overall expectation of global skin colours. So what this means is that they were saying, basically, if you assume what region is going to have what skin colour, then this was pretty accurate. Because they didn't have information on the skin colour of these individuals. So they would say, so for example, a sub-Saharan African is typically known to have a very dark skin colour, which is also the case, but not all New Guineans and blah, blah, blah. And they exclusively predicted dark black in the sub-Saharan Africans, which is the darker skin colour, and therefore it must be right.
I don't know what I think about that now.
No,
So this study agrees with stereotypes.
pretty much.
Yeah.
The sentence that they use is, their words is, although skin colour phenotypes are not directly available for the individuals analysed, they can be broadly assumed from general knowledge of global skin colour distribution.
Oh.
Which, this is where
Oh.
I had a Mean Girls moment.
Oh.
When there was a bit when Katie's being introduced to... Have you guys watched Mean Girls?
Some of it.
Years ago.
Okay.
Boys.
When Katie's being introduced to everyone.
Yeah,
and
yeah.
And,
I know
and
what
thing
you're
is
gonna
like, so if you're from Africa, then why are you white? Anyway, so this is pretty much what they're saying. And there's, another thing that they said is Europeans are known to have very pale and intermediate skin colour. And indeed, they predicted these 3 categories in the European regions. So they're not wrong.
They're kind of...
But-
it like pointing out the obvious while being a bit racist?
I don't think it's racist. I think it's a very big assumption and generalization that if
You're not
you have
supposed to assume, though.
yeah, and this is the problem, because if they're using this for forensics, which is what they're saying it's
Yeah.
for, and you're trying to catch this guy in like Africa, there could be a white guy committing crimes in Africa.
And then if you assume
It carries
he's-
that bias forward.
Exactly.
Yeah.
So yeah.
Does it say how they got their data set?
So these are from established genome projects.
Okay.
Yeah. So one of them is the 1,000 Genomes Project, which is this huge like global collaboration on trying to get all these genomes from people.
Because I'm wondering, because the biases associated with those data sets will feed directly through to those stereotypes.
Kind of and not kind of, because if you remember, we're looking at SNPs, and these are inherited through global like evolution and blah blah blah.
Yeah.
So potentially, but
Well, you know, if all the participants from the 1,000 Genomes Project were from only certain areas, you know, if there was certain areas underrepresented.
Yeah, but they've said that they've only got data from 59 regions.
Okay.
Oh, okay.
Yeah.
Yeah, fine.
Also, I think Planet Demelza's doing some fast googling.
There's the region. African.
They don't have any Canadian or
Yeah.
Russian
or
or Australian. Or very little South American.
None in Australia.
There's no like Aborigine or anything like that, or like
No.
the Pacific Islands.
No. So they are missing a lot, and actually, it's interesting that because a lot of these places that you said they don't have, so the Aborigines or Pacific Islands, they're going to have very like set.
Yeah.
There's not going to be much mixing going on there.
Yeah, there might be quite an isolated population.
Exactly. One of the things I was saying is that Asians are known to vary highly in skin colour across the region, with the exception of very pale skin. So they predicted all of them. They predicted all 5 categories for Asia. It was interesting, this paper, because I think this is actually one of the first papers I've read where they're trying to promote or like validate a product that they've come up with like this. So one of their results sections was actually just them telling us that they put it online, which is very nice. Thank you for that. And
Just
then,
a little plug.
and then they're giving you some tips on how to interpret their data. So if you use our program, this is what you need to do to be able to look at your results. So they're saying that because skin colour is so globally variable, when you plug in your genotype of the sample that you have, it's going to give you prediction, like probability values for the 5 categories that they have. And what they're saying is that you take the highest probability value as your main outcome, but then also you need to take your second one and think that it might influence that first one. So they're saying their system works, it's really good, and now they have this new way that they can identify skin colour as well when they have a DNA sample, and how useful it is in forensic investigations.
Was it self-funded?
It has funding support from the National Institute of Justice.
Oh, interesting.
Oh, that sounds like a comic book.
It does, right?
Yeah, Justice League.
Yeah.
So there's no similar products that compare skin tone?
As far as I'm aware, this is the first one. This was published really recently as well. So it was published this year, last month. So as far as I am aware, this will be the first validated forensic platform that they can have that has all 3. Yeah. But it's interesting because Violet Demelza here, she got 23andMe done. So she gave me some of her results that she's had through 23andMe, and she also plugged it into lots of other websites where you can upload, and they will analyze it with their data or like what they have, and it'll tell you just things. So Demelza has dark brown hair with green or hazel, don't really know what hazel actually is, we've realised, eyes.
It's closer to brown, Hazel.
Oh, okay, so green eyes, dark hair, and
Pale, but
yeah.
I feel like easily tanned.
Yeah, Demelza does tan really easily.
Yeah.
So from 23andMe, it's saying her skin pigmentation, she is most likely to have lighter skin, which is fairly accurate. It's also said that she's likely to have blue eyes with a good confidence,
Which is
wrong.
untrue.
She is most likely to have lighter hair as well, which she doesn't.
Also untrue.
So really, how accurate is this really? So yeah, I thought the interesting thing for me was how, when you watch all those crime shows and they make it look so easy to find out all this information, and they're in some high-tech lab, or they just type a few things into the internet and all this information pops up about you. But really, we can't even tell what bloody skin colour someone has, or eyes, or hair. So...
Yeah, I guess the reality is there's loads of people sitting there just scratching their heads thinking like, maybe.
So now when we're watching CSI, we can be like, they should be using this platform, or should they?
I don't know. Are you convinced? If you were, let's say you were top dogs in this big say you're top dogs of the FBI, would you use this software?
Criminal Minds.
You wouldn't use it in evidence in the court, but you use it to narrow the population that you're looking at. Why not?
Yeah.
It's easy, isn't it? But you wouldn't use it in court because it's so varied. You couldn't use it as evidence because it's flawed, isn't it?
Yeah.
I was going to say this. It's so kind of. I don't know if it's not necessarily probabilistic, but it seems like there's so much kind of variation between what we can say definitively and what this is saying it's like, oh, you might be this, and then it could be wrong. It's a bit like doing a lie detector test, or not the same, right? Because that's just wrong.
Yeah.
But there's
like, how can you trust it?
There's too much noise amongst the signal.
Yeah.
Yeah. What about you? Would you use it?
I agree with the noise.
Yeah.
It's just a bit.
Yeah.
Like, how could you ever be sure?
See, this is what I would do. My perfect team. So I would have this test, and then I'd bring in my little team from the BAU at the FBI, the Behavioural Analysis Unit. They'd come in, and then Bones, she would come in.
Is BAU the Criminal Minds?
Yeah.
Yeah. That's my
Fucking
favourite
love
one.
Criminal Minds.
That's the best one.
I need to catch up though on the last season.
Oh, I've never watched it in order. I love how they're all standalone. I sometimes just
Yeah, no.
flick by.
Was it you guys who were talking about? Sorry if this is going to ruin the thing.
Sorry?
Who were talking about Walking Dead?
Yeah.
Yes.
What was the really bad thing?
We can't say right now.
Okay.
Sorry, I'm kidding it.
Tangent.
Let's wrap shit up.
Yeah.
But yeah. Did you guys have fun?
Yeah, it was
Yeah,
great.
it's interesting. Yeah.
Did you guys learn stuff?
I did.
Yeah.
I did.
Are you interested in becoming scientists now?
I
Silence.
Ruled it out though.
The thing is, right, I feel like, okay, so this paper that I've done is such a perfect example of like, there's so much work that's gone into
Oh
this.
god.
Yes.
Yeah.
And then at the end of it, we're like,
Yeah.
But equally,
Relatively strong.
Alice's paper
Mhm.
was an example where so
So
little.
They just baked some bread, fed it to some sick women, and
See what happens, right?
thank you so much for coming.
yeah,
Thank
it's
you.
been great.
Thank you for having
Yeah.
me.
Would you come back?
Yes.
Yeah.
We are gonna need to just start
Recycling.
guests soon, so expect our call.
We're all running out of friends.
Oh, it was fun.
Well, I mean, Alice hasn't even brought any friends. So,
I don't have very many.
so final thing.
I don't have any non-science friends here.
Our babes, Co-Fresh. We know for sure that you know we exist. And what our podcast is about.
And you gave us a winky face in your tweet.
It was actually a tongue sticky out face.
Oh,
which
yeah.
is- That's
Oh,
even
shit.
naughtier than a winky face.
So naughty, and you haven't even sent us anything yet.
Yeah. You should at least send us some snacks first.
Yeah.
So do that, please. Thank you. We love you. Okay. Bye!
Goodbye.
You can say bye.
Bye.
They never normally say bye.
I know, but like, I just felt like they... I don't
That was
know.
a good bye though. It sounded like you were like singing. Like, bye. Very bad. Yeah, very like baritone.
Question, could you eat meat on the FODMAP?
Yeah.
Okay.
There's no fibre in meat.
I know. I'm ready.
Hang on a sec.
These are real good.
I quite like
Are you done? Oh my god. You're banned.
Now I'm really thirsty.